folks are starting to populate, I want to just give a say good afternoon or good morning. 11:30 is always that weird time. I'm not sure if it's afternoon or morning, but we're going to go with good afternoon to everybody. And welcome to this week's Business Unusual. As many of you know, my co-host Peter Klein and I started doing these weekly webinars right when COVID lockdown started. We felt it was a good resource to the business community to get industry leaders each week to talk about what's going on from a boots on the ground perspective with COVID, how it's impacting their industry, what they're seeing from a healthcare perspective. And it's been incredible. The feedback's been incredible. The response has been incredible. And our guests have been very candid. And as you know, we're not political at all. We want to get facts out to the business community so folks can understand how to gauge their response to this ongoing pandemic. Today, we're incredibly happy and proud that Dr. Bruce Stillman is going to be joining us from Cold Spring Harbor Lab. And Peter, I want to thank you. I know you have a good relationship with Bruce, and I know you want to give him an introduction, so I'll turn it over to you.
Thanks, Joe. Thanks for all your leadership on Business Unusual. And it's been 5 months that I can't believe is 5 months. It's been unbelievable. But we really, I really enjoyed the opportunity to be with you and weekly on these programs. And it is an honor and privilege to know Bruce Stillman and to be sitting on the association board of the labs. I've done that for the last few years, and it's a wonderful experience just from the people that are there and the amount of education we get from being on those on that board. So it's a wonderful organization, and Bruce will tell us more about its history. It goes back to the 19th century, and it is a quiet place in the sense that many folks don't really realize what they have there, and the brain power and the integrity of science is paramount. I'd like to introduce Dr. Bruce Stillman. Now, Bruce began his long affiliation with Cold Spring Harbor Labs in 1979 when he arrived from Australia as a postdoctoral fellow. He never went back, I don't think. He became a director of the Cancer Center in 1992 and president in 2003. Bruce maintains an active research lab working on RNA replication, a process of fundamental importance to life on Earth. Bruce's discovery of the origin recognition complex, he actually discovered something that is a scientific breakthrough called ORC, and his research on the process and regulation of chromosome replication have changed how scientists understand chromosomes. And for those of you going back to your high school biology, those are like genes. If they're not, maybe that is what a gene is, a chromosome. I think they're the part of the cell that's genetic material, but Bruce would know better. Bruce has received numerous awards and honors for his research, including the prestigious 2019 Canada Gardner International Award. And then this year, we're very proud to announce, in 2020, he's won the Dr. H.P. Heineken Prize for Biochemistry and Biophysics. Please welcome Dr. Bruce Stillman. Hi, Bruce.
Thanks very much, Peter, and thanks, Joe, for the opportunity to kind of participate in this series. It sounds like an interesting series that is probably the new normal in how we interact with our colleagues. I've done a number of these events with various clubs and organizations around the country, and it's getting a message out about Cold Spring Harbor Laboratory, which and I want to thank Peter for his support and enthusiasm about the laboratory, which he's one of our great supporters and friends of the laboratory. So thank you. So it's a pleasure to be here. Maybe I'll just start off by telling you a little bit about Cold Spring Harbor. As Peter said, we were founded in 1890 as an educational institution under the Board of Regents of New York State, really to teach science. And in 1900, Mendel's laws of genetics were rediscovered. Mendel published this work in 1865, but it was kind of ignored for a long time. And then three scientists rediscovered Mendel's laws of genetics in 1900. And that's when Cold Spring Harbor Laboratory became the first institution in the United States to do genetics.
Hey, hey, Bruce, real quick, is Mendel the monk with the peas?
Yep.
If I'm remembering my my
Hey, you, Joe.
You,
the monk
very
with the
good.
peas, right? Yeah. All right, I'm
Yeah, so
following you. I'm good. Yep.
He was an Austrian monk who had this fantastic garden and studied inheritance in plants, particularly peas, and was one of the most important discoveries in the history of biology. And it came out just 6 years after Darwin published his monumental work about natural selection. So that's an interesting thing, because my predecessor at the laboratory, who was director of the laboratory in 1900, decided to focus research at the laboratory on understanding Darwinian evolution in Mendelian genetic terms. And that led to a real revolution in genetics. By 1908, hybrid corn was developed here by crossing various strains of corn. The current and high-yielding corn was developed at Cold Spring Harbor in 1908. It led to a revolution in American agriculture. And we've been doing genetics ever since. There have been 8 people who have won the Nobel Prize have worked here, mostly in genetics. And the current laboratory has 1,100 employees, 600 scientists, an annual operating budget of about $190 million. And about 50% of the research income comes from our scientists writing grants from the National Institutes of Health or the National Science Foundation. And the rest come from endowment and fundraising and other sources of foundations. And we actually have 6 divisions of the laboratory. So we have a publishing division. We have a large Cold Spring Harbor Laboratory Press, which is headquartered in Woodbury. We have a conference center in Lloyd Harbor called the Banbury Center, which is like a think tank for the life sciences and medicine. We have the main research campus and a research campus in Woodbury in the research division. We also have one of the largest conference centers in the life sciences in the world, headquartered on the campus at Cold Spring Harbor. In normal times, we have about 9,000 scientists a year from all around the world visit Cold Spring Harbor to attend conferences here. We also have a conference center in Suzhou in China that we operate. And we are a degree-granting institution, so we have a graduate school. And our research is focused on cancer. We're a National Cancer Institute designated cancer center, the only one on Long Island. So we work on cancer, neuroscience, plant biology, and quantitative biology, which is computer science related to biology. And
DNA Learning Center too, right, Bruce?
yes, and then probably the one that's most impactful for the local Long Island is the DNA Learning Centers. We've started DNA Learning Centers. We own a school in Cold Spring Harbor where we teach science to many, many school districts across Long Island. We also have a second one in Lake Success, and we're building a very large one in downtown Brooklyn. We have one in Harlem. And then we've initiated DNA Learning Centers with universities across the United States and actually throughout the world, including Australia, China, and countries in Europe.
Hey, Bruce, let me ask you, so Peter and I are Stony Brook University, proud Stony Brook University grads, and you're talking now, and I'm digging back into my my brain, and I'm remembering Watson and the Double Helix was a book that I studied when I was in university, and that's Cold Spring Harbor scientists. And was that the discovery of DNA? Is that what occurred there?
No.
No, Jim Watson, who's actually still alive, he's 92, he and actually lives on the campus here. He and Francis Crick proposed the structure for the double helix in 1953. They worked at Cambridge University. Jim is an American, but he worked at Cambridge University in England. And actually, the connection to Cold Spring Harbor was that Jim came here when he was a graduate student in 1948. Then went to the university when he was doing a PhD in Indiana, and then went to Europe to do a postdoc, and that's where they just made that discovery. And Jim came back here in the spring of 1953 and gave the first public presentation of the double helix at Cold Spring Harbor at one of our meetings. So, and then he came here in 1968 to become director, and was director from 1968 to 1993 when I became director in 1994. And
so the double helix, which I know is the name of the annual dinner you have, fundraising dinner.
yeah,
The double helix is a structure that's in every nucleus that essentially is how the genes interstate. What's
so you know, Mendel discovered that genes are inherited in units, you know, that you can actually, they're mathematically predictable. And that was his big discovery. And then in the early 1900s, scientists, including scientists at Cold Spring Harbor, showed that genes are on chromosomes. But the discovery of the double helix, the intertwined double helix, in fact, I have one right here on my desk, so you can see one. That double helix explained two things. One is it explained that the bases of DNA were actually the units of inheritance, and there was a code. There are 4 bases, and the sequence of the DNA actually has a code that would tell the cell how to make proteins, and proteins are the building blocks of cells. The other thing is it also explained how the 2 strands could come apart and they self-template and they copy each other, and that's what actually I work on, how this molecule is copied. Every time a cell divides, you have to have an equal number of chromosomes copied from one cell to the next. And so it outlined how roughly the chromosomes are inherited, but over the past 30 or 40 years, my lab and others have figured out exactly how this happens, like down at the atomic level. And the final thing that double helix pointed out was how mutations occur. So an error in the copying process can cause a mutation. And this really opened up understanding of the gene, understanding of mutations, and understanding of inheritance. So it was really the beginnings of, 1953. That was the year I was born, so it was a good year to be born. So I kind of grew up with DNA and did work on it.
Bruce, I'm just wondering, how did the pivot from genetics, or maybe it wasn't necessarily a pivot, but maybe it was just a movement towards oncology and cancer research? How did that come about? Because I mean, Cold Spring Harbor is known for its cancer research.
Yeah, so it was recognized in the beginning in the 1960s that cancer was, there were changes in the chromosomes in cancer cells. And in the 1970s, including work that was done at Cold Spring Harbor, there was a realization that changes in the DNA inside our cells in our body, the so-called somatic cells, the cells that make up the bulk of our body, errors that accumulate in the DNA could contribute to cancer. And so by 1968, when Jim Watson came here, he figured out that to understand cancer, you really needed to understand the genetics of cancer. And we became a leading center in the world in understanding the genetics of cancer. In fact, the first human cancer-causing gene was cloned here in 1981 by Mike Wigler. And there are many, many major discoveries in the cancer genetics field that were made at Cold Spring Harbor, some of which led to Nobel Prizes. And so cancer is a genetic disease. It can be inherited, like the famous BRCA1 breast cancer gene, that you can inherit the susceptibility to cancer, high incidence of cancer from one's parents. However, for the most part, and for most of us, cancer is the accumulation of mutations in our cells in our body that cause those cells to proliferate out of control.
Hey, so Bruce, we're going to talk a little bit about the work you guys are doing as it relates to COVID, but let's go back to March for a moment. Okay, so you're a scientist and you're a CEO, so you have to run the lab,
Yep.
right? But I'm sure you have your it must have been a very challenging time. So I want to roll the tape back to March, and the governor says that we're going to be shutting down businesses in the middle of a pandemic. So what is that like for a CEO who's a scientist, right? So we spoke to Ken Kashansky from Stony Brook, and you know, it freaked him out, and his brain went in all sorts of directions. So I'm curious as to what you were personally going through at that time and how you viewed where we were at.
Yeah, it's interesting. Yeah, I know Ken very well. Actually, I'm going out to Stony Brook, since you're a alum. I'm going out to meet the new president this afternoon, so have a discussion with her. We actually were a little bit prepared for COVID because we knew about it probably before anybody else in the United States, because certainly on Long Island, because as I said, we operate a conference center and teaching laboratories in Suzhou in China. And I was actually scheduled to go to China, to Suzhou, in February. And that got canceled, obviously, because of the pandemic in China they were right in the middle of everything in February. So our employees were sequestered at home in January of this year. And we were dealing with the pandemic in China through our employees there, even as far back as the middle of January. And they were sequestered for 2 months, and in the middle of March, when everything started hitting in New York, they were actually allowed to start to go back to work in a limited way. So we were dealing with this, and so we kind of anticipated what was going to happen here. And we very rapidly put in place a number of measures at the laboratory. First of all, we worked with the governor's office, and we have a very close relationship also with Northwell Health. And Mike Dowling was a principal advisor to the governor on this entire COVID pandemic. And Northwell was and Stony Brook were kind of, you know, the center of the infection in New York, which was, as you know, was the highest in the country and in fact in the world in March. So we immediately repurposed about 7 of the laboratories here. Most of our employees were working from home using Zoom. These were the administrative employees. Our scientists were able to work in the laboratories. We have an unusual campus. We have a very distributed set of buildings. And so we were able to spread out our scientists into laboratories that were normally occupied for teaching purposes in the summer, and spread the scientists out. And we repurposed about 7 of the laboratories here to work on COVID, and often in collaboration with either Northwell or other centers. So some of our scientists, for instance, purified virus proteins that were then shipped off to various places and used to develop antibody tests for the virus. We have worked on with clinicians, and in fact have done clinical trials on treating the most severe symptoms of COVID, which is the respiratory distress syndrome, why patients have to be intubated. And we've discovered, along with scientists at the University of Utah and Weill Cornell Medical School in New York and Northwell Health, we discovered that the severity of the COVID infection, of the SARS infection, and the most severe symptoms are associated with a hyperimmune system that produces from cells called neutrophils, which are part of our innate immune system. It produces these things called nets, which are part of the normal process to fight bacterial infections. But what happens in COVID is this goes completely haywire and is responsible in part for the severe respiratory distress and also the blood clotting that occurs. As a result of that insight that was made by one of our scientists, a woman named, professor named Mikola Egerblad, that has now gone into clinical trials to treat these overreaction of neutrophils in the most severe patients, and there's clinical trials going on throughout the world now. In another area we worked on, there was a lot of interest in repurposing, looking at repurposing existing drugs and seeing if they were effective. You know about the famous hydroxychloroquine that the president touted. Then there was remdesivir, which was the Gilead drug that was originally developed for Ebola, which has a kind of a modest effect, I would say, but a demonstrable effect. And then we got involved with Northwell in a clinical study and research on a drug called famotidine, which is famotidine.
This is a great story. This is actually a great story about a researcher that was in China, and he came back and he was talking with one of his friends at Cold Spring Harbor.
Well, there's a scientist who works at, or a clinician who works at Massachusetts General Hospital, and he was actually in Wuhan in December and January when the virus broke out in Wuhan. And they noticed, the Chinese doctors and he noticed that people that were taking a heartburn drug, Pepsid, had less severe symptoms than people who were not. And he came back and reported this to the US Department of Health. And the Department of Health asked Northwell Health, because it was the center of infection in New York, to run a clinical study on famotidine, that's the active drug in Pepsid, whether that might have an effect on the most advanced patients. And that trial got started and actually was complicated because of the use of hydroxychloroquine. And in fact, it's been a very complicated trial. It was done on the patients that were intubated and in hospital. But we have a number of clinician scientists at Cold Spring Harbor. These are people who have MDs and PhDs. And one of our clinician scientists, his sister got infected. She works in the health department in New York. And she got infected and was quite severely infected and was on her way to the hospital. And our clinician scientist said, before you go to the hospital, why don't you just take this drug and see how it works? It was famotidine. And he treated her for a couple of days and she got better. And as a result of that, about 10 other people in the health department in New York who were also infected were similarly treated and monitored by our scientists, clinician scientists, for the effects of the symptoms. And all of them got better. And that clinical study, it's not a clinical trial, but that clinical trial of just 10 patients was published in a journal, clinical journal. And that's led to us and Northwell proposing to the FDA to do a study on the use of famotidine in outpatients, not the patients in hospitals, but in outpatients, just as they were treating the people in New York City. And that has been submitted to the FDA for approval which is going through a little bit of regulatory stress at the moment because the FDA are very, they've been very cautious about what they approve and what they don't approve for COVID now, given all of the politics behind all this. So we are going to do this trial, and we've raised the funds to do it, but we're just waiting for FDA approval for it.
It seems odd, doesn't it seem odd to you, Joe, that something dealing with breathing, COVID, and then heartburn? It's almost like incongruent.
Well, yeah, the heartburn is explainable because we think originally it was thought by a number of people that the drug blocked the virus replication, but that was shown by our scientists and others not to be the case. The drug targets the control of the body's physiology through molecules called histamines. And these antihistamines, which is what peptide is, it controls a whole bunch of things in the body, including blood pressure and blood flow, but also the innate immune response. And what happens in COVID, the coronavirus, there are many different coronaviruses, and they've been around for a long, long time. And many of us have been infected with previous coronaviruses, and fortunately, most of them are actually not very severe. This one actually happens to be extremely severe. And the reason why it is severe is that when the virus infects somebody, it stimulates, it shuts down the what's called the interferon response, which is a natural antivirus response. The first response to a virus is called the interferon response. And then there's a second response that the immune system triggers, which is the release of a whole bunch of immune regulators. They're called cytokines. And these go around the body and trigger the immune system to fight the virus. But normally, most viruses, like measles and flu and all of these other viruses, they shut that down so the immune system doesn't attack the virus. What SARS does, and is very, very unusual, is it actually induces huge amounts of these immune regulators and keeps them going instead of shutting them down, which most viruses do. This actually stimulates them to go around the body, and that's you might have read in the popular press about cytokine storms. Well, that's what this is. The virus induces a massive immune response in the body, and the body doesn't know how to handle this. And so different people respond differently to the virus, and elderly people in particular, or immune compromised people in particular, or people who have comorbid conditions, diabetes being one of them, have an abnormally high cytokine response or these immune regulators, and the immune system just goes completely out of whack.
Yeah, and Bruce, I want to thank you for saying that, because I think in this super hypercharged environment we're in, right? At least I've studied this very closely, just don't have clarity, and that was a very clear explanation, I think, as to why COVID-19 is such a high risk. You know, we hear about H1N1. So explain so we understand the difference between what H1N1 virus was and what COVID virus is, and why we're in this sort of, you know, part of our history where we're masked up and socially distanced where we weren't in the past, right? Because I think there's clear scientific reasons, but because of all the noise out there, we don't get to hear them.
Yeah, well, you know, there was a lot of denial of the effect of this virus throughout the world, actually, including in China. In the original people, the first people in Wuhan who recognized the infection actually raised concerns about this as a very dangerous virus. And in fact, the principal physician who made those calls to the Chinese government actually ended up dying of COVID. But the central government ignored it for a period of time. And it was only after it became a real serious problem. So the virus had a chance, unlike there was a previous coronavirus outbreak in China in Guangzhou in 2011. That was the SARS-1. And SARS-1 actually made it to Vancouver, but it was very, very rapidly shut down by limiting transportation. And that response, and it really kept the virus under control. This one got out of control for too long in China. And then there were people who made it into Europe, and the virus in Europe mutated a little bit and changed a little bit, and actually we think became much more infectious. And that virus came to New York. And so the virus that was in Italy and New York, remember we were the big centers of virus infection in March, that was a slightly different virus than came out of China. And we believe it was more, well, it's been shown to be more infectious. But unlike the flu, these viruses come out of animals. Flu comes out of birds for the most part, and the genomes recombine to produce new viruses that have never been seen before. I mean, they're in the class of, say, flu or coronavirus in the case of SARS. They're new viruses that the immune system has never seen before, and they can cause devastating effects. And H1N1 is one. The Spanish flu in 1918 was a big one. Hong Kong flu in 1968 was another one. They tend to come out of China because that's where the viruses originate. And for flu, we understand a lot about that. There's a somewhat of a prediction. Occasionally, with the annual flu shots, we can predict what type of flu viruses are coming down the road. And there is a vaccine for flu and some anti-drug treatments. For coronavirus, there was really nothing. And I'll mention a little bit about what therapeutics are coming down the pike, but there was really nothing. And so this virus also been very unusual in causing the cytokine storm, really for vulnerable people who could not deal with this immune challenge. This is one of the reasons why it's caused so many deaths. And it's still a very severe infection, even in people who are not hospitalized. You can still have a very severe infection. And there's also beginnings of evidence that it may actually have long-term effects, for instance, on the heart and other organs because of these reactions. When you have such a very, very heavy reaction to a virus infection in the lungs and the heart and the kidney, you can get cells that change and they become what we call fibrotic. And these fibrosis type things can actually cause long-term medical problems, as they are known to do in other diseases. And so I don't think this is a virus that should be taken lightly, and hence the need for social distancing and wearing masks, which is actually proving to be very effective. I think New York has done a remarkably good job in shutting down the initial peak and then keeping it fairly low. We did not see that in the rest of the country, and you're seeing the consequences of it. So now, as we all go, as kids go back to school, and I think we'll start to see a little bit of a resurgence. However, I think there is some hope in the near future, in the very near future, that there'll be some therapeutics that will be available. I mentioned the trial that we're doing on famotidine, which will not prevent the virus infection, but limit the symptoms, if that proves to be the case. in a blinded clinical trial. The other thing is that a number of companies, including Regeneron in New York and Eli Lilly, are producing what are called monoclonal antibodies. These are antibodies that are raised against the virus proteins themselves that can block the virus from infecting cells. They're called neutralizing antibodies. And it's possible to identify these, clone the genes for them, and then put those genes into cells and make those and have those cells produce extremely large amounts. Monoclonal antibodies are actually one of the most commonly used drugs available today. Drugs like Herceptin for breast cancer, Enbrel for arthritis, and all of these. These are monoclonal antibodies that are made in huge vats of cells that produce these drugs. So Regeneron and Eli Lilly are now in clinical trials, in very advanced phase 3 clinical trials, testing the ability of these monoclonal antibody cocktails to prevent virus infection and also treat virus infection. And there's a lot of hope that an expectation that those will work. They worked in chimpanzees or in monkeys, and they worked in animal models of the coronavirus. And in early clinical studies, they've shown to be effective as well. So definitive clinical trials are going on now. I anticipate that they will actually start to have results in the next few weeks. And the National Institutes of Health and the Department of Health and Human Services have actually worked with these companies to make very, very large amounts of these antibodies so that if the clinical trials are as expected and they're positive, then they'll be readily available.
What is the risk of mutation that you see? So, you know, I know with the flu shot every year, right? And I do a lot of work with some of the different pharmaceutical companies, and they're all racing to get the right strain that they think is going to give the best flu shot because the flu changes. And you mentioned that COVID mutated a little bit in Europe. I mean, what's the risks there, and how is that going to impact the vaccine that comes out?
Yeah, so good question. So influenza virus is one of the viruses that mutates the most. It actually mutates and changes enormously from year to year. And it also has the ability, it has quite a number of different genes on different molecules, different chromosomes inside the virus. And these can recombine. And so this is how you get new strains of flu. That doesn't happen for SARS. SARS is actually a relatively stable virus. It does mutate, but it's relatively stable. And the therapeutic antibodies that I mentioned, Regeneron, for instance, have made multiple antibodies against the same virus protein, the so-called spike protein on the surface of the virus. They made a cocktail of multiple antibodies so that the likelihood, you could get a mutation against one, but the likelihood of getting a mutation against two that would block both of them is very, very, very low. And so this cocktail of antibodies is probably going to get around the mutation. Now, with a vaccine, so these monoclonal antibodies have to be injected. They will last in the body for about, the experience with other viruses is they last for about 2 to 3 months. And this will be sufficient, we think, to enable us to wait until the vaccines have been proven to be safe. And this is a very, very important thing for coronavirus. There's a lot of pressure to get these vaccines out there, but previous vaccines against other coronaviruses, like the Middle East infection in about a decade ago or so ago, and the SARS-1, which was 2001, I think it was, those vaccines turned out to be not effective, and in fact, in one case, a vaccine actually enhanced infectivity. So these vaccines that a lot of companies are developing have to be proved to be safe, and it's really, really important that they be given time to do that. And so I think the use of these therapeutic monoclonal antibodies will, and even prophylactic, meaning preventing infection, will give us some time to then wait for the definitive clinical studies on the vaccine itself.
Bruce, am I reading or listening to you correctly? You sound optimistic, if I should use that word in quotes perhaps, that the fall this year will not be certainly as bad as the spring was. I mean, are you sensing that?
Yeah, and I think so. Well, first of all, we know a lot more about this virus than we did in March. We know how to treat patients who are infected much better. We know about all this cytokine storm business. We know how to mitigate the effects of the cytokine storm, and there'll be, as I said, there'll be other ways of dealing with that down the road. So, you know, back in March at Northwell, I mean, everybody was just, they did a remarkable job, and so did Stony Brook, in a remarkable job in dealing with these patients. I mean, they suddenly had to increase the number of ICU beds enormously, and create effectively, Northwell built effectively 2 new hospitals within weeks, and to deal with all of the patients. And there were a lot of hospital beds that were repurposed as ICU beds. I don't think that's going to happen in the future, but also I think that these therapies, particularly the monoclonal antibodies, if they work, and I'm pretty optimistic that they will, then they will be available to the people who are the most susceptible, elderly people, people with comorbid conditions, and maybe even more generally in the general population, if they can produce enough of these. Regeneron signed a deal with Roche. Roche has got the biggest capacity in the world for making monoclonal antibodies as therapies. And they're gearing up making those now. So I actually think that these antibodies, if they're going to be widely available, will mitigate the effects of the virus in the fall. And, you know, we'll just have to wait and see if there's going to be enough of them, and if they're effective, first of all, and if there's going to be enough of them. But I think we'll hear within the next few weeks whether they're working. The clinical
What
trial.
risk is there on black market stuff, right? So I hear you talking about antibodies, and if there's going to be enough, and my mind goes to black market drugs. Is that something we should be concerned about?
Unlike these other drugs, I mean, unlike a lot of other drugs, where they're produced and there are stores of them, like, you know, small molecule drugs, and people have kind of cornered the market on these and had a black market. That will probably not be possible with this, because these monoclonal antibody drugs have to be produced in a particular way. The technology to produce them, nobody else can produce these unless you have the huge facilities that Roche and Regeneron and other companies have, Eli Lilly. And they will be produced in hopefully very large amounts. And then the question is, how are they going to be distributed? And I think the companies will distribute them directly to hospitals and to clinical centers, which is the way they're not like a drug that you can sit on the shelf and, you know, store in your bathroom and things like that. They're not that type of drug. They'll have to be administered by a physician or a nurse. And but, you know, they're very widely used drugs. I mean, monoclonal antibodies are actually very commonly used drugs nowadays. And I think there'll be, and also there's going to be a lot of visibility around them as well. So I don't think there'll be a black market on them.
Yep. Hey, Bruce, let's talk about you for a minute, right? So I'm curious as to your journey here. You know, for me, I grew up in an Italian, Irish Catholic house that argued a lot. So me being a lawyer was a natural progression, right? So how did you get interested in this, and sort of how did you get to where you are today, right?
Yeah, well, I grew up in just outside of Melbourne in Australia, in what then was a kind of a semi-rural area, but is now an outer suburb. We were completely surrounded by orchards and things like that, so it was kind of a good place to grow up. When I was 11 years old, friends of my parents, he was in a civil defence rescue squad in Australia. This is a volunteer kind of rescue squad that would go out and search for people who were missing in the Australian bush, or go and fight fires or things like that. And they had a cadet program, and he encouraged me to do that. So at the age of 11, I joined this thing, and I found I had an enormous affinity for liking medicine. I started reading everything I could, encyclopedias of medicine, and by the time I was 16, I was so good at it that I won national championships in emergency medicine and first aid. And so I kind of decided I wanted to do medicine at university. So I went to university, and when I was there, I realized that I actually didn't want to really practice medicine, because I'd seen enough of that in hospitals and things like that, that I really wanted to do something effectively and do medical research. And so I switched over and started doing research. Had by then got an interest in cancer, on viruses that cause cancer. And so I went to the medical school at the Australian National University in Canberra, did a study in medical microbiology, studying viruses that cause cancer, and I made some important observations when I was a graduate student. And that kind of intrigued me a lot, so I decided I was going to focus my initial career on that area. And so Cold Spring Harbor happened to be one of the strongest places for understanding the genetics of cancer, as I said before. And Jim Watson came here in '68, so I finished my PhD in 1979. And I wrote to him and asked if I could come and study. Had this idea that I wanted to study, and he said yes, and so I came here. And initially just for 2 years, but things went very well. I got promoted to the staff, and then met my wife, who's from Long Island, and that was it. I'm kind of got stuck here. So
how lucky are we?
Yeah. So no, I actually enjoyed it. I mean, I go back to Australia a lot, and I consult with universities and colleagues and companies in Australia, but I like it here.
What's a typical week like for you? So one of the frustrations I have as the managing partner of my firm is, you know, I actually like working with clients and on matters, but administration of the firm gets in my way sometimes. And so I'm curious how you manage that, and what's it like managing one of the top, labs in the world, how that works.
Well, it's actually quite an honor to be president of Cold Spring Harbor Laboratory. It's we were rated as the #1 institution in the world in productivity in science and medicine, and have been for quite some time. And it's and the laboratory through our conference centers and other programs have a very, very big impact on world science. And so it's good to be an active scientist as head of Cold Spring Harbor Laboratory. One of the things that enables me to do it, I still run my own laboratory. One of the things that enables me to do it is, well, there's a number of things. One is I have an incredibly understanding wife who's good, so she lets me work all the time. The second is that we have a system here. I have a chief operating officer of the laboratory, a guy named John Tukey, who's really fantastic, and he runs the administration at the laboratory. So basically the COO is the person who is the operations person that does all the budgets and things like that and oversees that. And so we're kind of almost a team rather than a, CEO, COO type. And that helps an enormous amount. And then we have great supporters, and Peter's one of them, but we have a great board of trustees. We have a Cold Spring Harbor Association board that Peter's on, and they provide a lot of support and help with fundraising. So a lot of my work is focused on fundraising, and that helps to have good people working with you. So you can do it. And the final thing is I live on the campus, so I spend most of my time here, and especially now since I can't travel. And you just do what you have to do. So I work a lot, but that's the way.
Do you think in part, Bruce, do you think it's also the fact that the folks around you, the top tier scientists around the world that come to Cold Spring Harbor, it sort of ups your game to a sense as well? Everybody is sort of top tier, so it's sort of like...
Well, one of the great things about Cold Spring Harbor Lab is the scientists who actually work here get exposed to the world scientists by, we normally have about 9,000 visitors a year. So you get to interact with pretty much everybody who's world leading in science. They come to Cold Spring Harbor and talk about their science. And I got to meet, when I was a younger scientist here, I got to meet some of the world's very prominent scientists, including Francis Crick and lots and lots of others. And I got to know these people really well. And you kind of get used to having really, really bright people around, and that's a great thing. And so that environment creates a lot of intellectual cross-talk. And so some of the ideas that I've had in my career have come from talking to scientists who have visited here. And through those conversations, I've got some ideas that we then have executed down the road. So it's really like a Grand Central Station for exchange of ideas.
Yeah, we got a question about internship opportunities. So at the lab, is it that each scientist drives their own internship programs? Or are there internship opportunities at the lab? How does that work?
Yeah, so we have at multiple different levels actually. So the way the research works is we have faculty who are assistant or associate or full professors. We have about 57 different laboratories. In those laboratories, for instance, in my laboratory, I have 10 people who work for me, either postdocs or students or technicians. That's the typical structure of the 57 labs at Cold Spring Harbor. We can take, we do have an undergraduate program, summer undergraduate program. We didn't do it this summer, of course, but normally we take around about 25 undergraduates from all around the world who come and do a summer program here. We have, through the DNA Learning Center, we have opportunities for high school kids to work in the DNA Learning Centers to help out. And then we have a thing called the Partners for the Futures program. This is where we take 14 high school students who come and work at Cold Spring Harbor full-time for a year while they're at high school. Now, these are kids that have usually gone through the DNA Learning Center programs, have really got interested in science, and have really excelled. And their teachers, by the time they're in 12th grade, they're kind of bored to tears at high school because they're really, really good. And they tend to come and work here because the last year of high school, they've done everything that you could possibly do. And so they work here and they get a lot of experience in research. So we have those training programs. We actually teach courses. We have summer camps in the DNA learning centers. So the DNA, even this year, we did virtual summer camps. And so there are programs there where students can go and do, fairly sophisticated research in the Learning Center laboratories. So there's a lot of opportunities for training and internships that we have.
Yep. So let's end with sort of a shameless plug, right? So fundraising's important. What are some of the ways the business community can help support the lab? Because I think anybody watching this who is going to watch this realizes how critical the lab is, not just to Long Island and not just to the world, but to humanity, right? It's to humanity, the world, the work that you guys are doing. So how can the business community help support?
Well, there's a couple of ways. We do have a corporate advisory board made up of representatives of corporations on Long Island. And they meet and they support, mostly through an annual golf tournament, support the laboratory. The board that Peter's on, the Cold Spring Harbor Laboratory Association, this is really the organization that is the friend and fundraising arm of Cold Spring Harbor Laboratory. And by joining the association and becoming active in the association, attending, we offer public lectures, we have concerts, we have all sorts of things under normal times. One can come to the campus and participate in these events. We've been doing virtual public lectures during this year. The Cold Spring Harbor Laboratory Association is a good way to get to know the laboratory. You get a lot of information and it's also a way of a vehicle for donating towards the laboratory. And then people who, like Peter, who's got really involved, get to work and get to interact with our scientists and learn a lot more about the research programs here. And Peter's met quite a few of our scientists and got to know them. And that leads to either, in Peter's case, or in foundations, or in or recommending other places of support of the laboratory. So we do fundraising through kind of networking.
You also have the annual Double Helix, which is a
yes, and we have a big fundraising dinner in New York, normally in normal times, every November, which raises around about $4 to $4.5 million in one night for the laboratory. We have a lot of incredible support from our trustees and association members. So there's a number of events we have throughout the year to raise money, golf outings and a Women in Science event and things like that. But people find their level of comfort in how they interact with the laboratory, and our experience is that when people find out about what's going here, they tend to latch onto things. And some people, I mean, there's a lot of people here who are interested in what we do because of what we do, diseases that have affected them or family members. And that's resulted in some cases, Cold Spring Harbor Laboratory actually starting new programs because we've worked with local foundations.
It's terrific. So we're out of time, Bruce, but what we do is always give our guests the last word, and it has to be something positive and optimistic. So that's the rule, because you scientists can scare the hell out of us sometimes.
Yeah.
So,
well, I think one of the things I think a lot of people have realized out of all of this is that science actually really can matter and have a big impact. And I think the global collaboration that's happening around the world in response to the pandemic, everybody thinks it's been a horrible year. It could have been a disaster. Absolutely. If we hadn't had science. I mean, this virus was cloned within weeks of it being, the genome was cloned and sequenced within weeks of it being isolated. And as I said, we've got to the stage now where there'll be probably therapeutic antibodies. So if we didn't have the science that backed all of that up, if you think of all of the things that you've had to have discovered to be able to get to this stage of dealing with the pandemic, and the science is supported in this country, and the reason why the United States is so predominant in the global impact in science and has been for a long time in terms of number of Nobel Prizes and the number of patents and the impact, is because of federal government support through your taxes and also through philanthropy. That really, the United States invented philanthropy, or not kind of really, but the Medicis did it back in Galileo's time. But John Rockefeller really initiated the modern philanthropy era in the United States. And the United States, the reason why the United States is so predominant is through philanthropy supporting science. So I think we'll move forward out of this pandemic within the next 6 months. There'll be therapeutics, I'm pretty sure, and there will be still a need for caution because I don't think everybody in the world will be able to be treated with these therapeutics until there are widely distributed vaccines, and then they'll take a while to distribute around the world. So I think travel is going to be slow coming back, but I think we've found through this vehicle we're on now, we've found a new normal for doing business. And I actually think the economy will come back fairly abruptly because of that.
Yep. Peter, that's your segue, right? The market is going gangbusters again, and nobody knows why.
So let me say that I'm optimistic from a perspective of being involved, and it's a privilege to be involved in the Cold Spring Harbor Labs, and just having that kind of organization and brain power and commitment to science and humanity in our backyard here on Long Island makes me extremely proud to be a Long Islander. So I am optimistic in the furthering of scientific discovery, and I suspect a good deal of it will come from the folks at Cold Spring Harbor Labs.
Yeah, and that's great. And I'm optimistic because what the hell else am I going to do, right? You just have to be optimistic. We got to move through this. We got to move through this together as safely as we possibly can. Dr. Bruce Stillman, thank you so much for such a great segment, so much great information there. And the work you guys are doing at Cold Spring Harbor Lab, I am sure the business community recognizes that and will support it. Folks, we are off next week. We're going to take the day after Labor Day and try and get the cobwebs out of our head and get back to business. But we'll be picking up the week after that. Bruce, thanks so much for everything. Keep up the great work. We really appreciate it.
Thanks, Bruce.
Thanks, Joe. Thanks you for